So do we start, I guess it's almost 1:45. I think the scheduled starting time is 4:30, yes? 4:40, okay. So we can start any time. Can I expect that most of the people are here?
I think yes.
Okay.
The microphone is not present.
Okay. Okay, can you hear me?
Sorry.
Okay, can you hear me now? Is that better? Okay. So anyway, it seems like we're a small group. So maybe if most of you is close, please, I encourage you to move close. Okay, so first, when deciding what to present, I have to say that I'm still somewhat undecided because I don't know enough about the audience. So maybe I can ask you guys just to give me an idea what your background is in biology and in biophysics, and what you would hope to learn from the course in the 3 days that we have. First of all, is everyone familiar with some biology, let's say, at the level of first 2 years? So a little bit of biochemistry or molecular biology, cell biology, or no? Or are there people here with no biology beyond just first year of general biology? Okay, and then do we have people who have had biophysics background? Okay, so it looks like we have a little bit of a mixed group. So what I've prepared, and we'll see whether or not this is suitable for you guys, is basically a 2-part presentation. So we'll do the first part today and then the first bit of tomorrow's session. And then the second bit of tomorrow's session will start on the second part. So the first part, I'm calling molecular biophysics. Biophysics of macromolecules, proteins, nucleic acids for the most part. And then the second part, I'm calling systems biology. So I'm not sure whether this is the title that I chose or whether it somehow got into the program, from molecules to organisms. I'm not really qualified to talk to you about organisms, but with the systems biology, we'll at least get to learn some approaches to modeling biophysical processes at the level of the cell, the organs, or the organism itself. So let's start out with the molecular biophysics. So I'm going to start out with a historical prelude, go back to the origins not only of molecular biophysics, but molecular biology itself. I think this is something that is important for us to understand, that it really came from physics, because that I think is an indication of something that's still true, which is that biology and physics are very closely linked. Although sometimes they're taught as if they're almost polar opposite subjects, but actually they're very closely linked. And So then going on to the history, we will see that in the beginning of molecular biology, there was the central dogma, which is something very simple, and it's still very much at the foundation of the way that people think about molecular biology. But the reality of how things work has now we know it's much more complex. So I'm going to talk a little bit about that, the original central dogma and a kind of updated central dogma. And this will bring us into the mechanism of the storage and transmission of information. Information, storage, transmission, transduction is very much at the foundation of biology. In fact, some people define biology as information transduction. So, but of course we don't have time in this series to cover that in detail, but we will just talk about the mechanism, the classical mechanism of the storage and transmission of information in DNA. We'll go through that very quickly. And then the protein folding problem. And depending on how confident you guys feel, there's just a little bit of the basics of protein structure that we all need to have a foundation in. And then when I talk about biological function of macromolecules, this is really going to bring us into the interaction of biomolecules with each other. Because actually the protein folding problem and the information storage and transmission problem are not the real point of molecular biophysics. Molecular biophysics is something that's very highly dynamic. It's about this dance, this molecular dance, these molecules interacting with each other. And one of the reasons I was insisting on using my computer, I want us to get just a little bit of a chance to hands-on look at some molecules and molecular graphics. So just introduce that in this lecture. There's a program, open source program, that I would like you guys to download this evening. And then maybe tomorrow we'll have just a very quick session actually looking at some molecules with our own hands. Okay, so to begin with, I want to address how, as physicists, we should be thinking about biology. Because I said, these are not polar opposite subjects. These are very closely related subjects. And the main point here is that organisms are made up of cells, and the cells have quite complex structures. They have organelles, they have nucleus, they have these structures called mitochondria, which are like protein factories, and structures with funny names like endoplasmic reticulum and so forth, which you can see under a microscope. So it's quite complex, but all of these components arrange themselves. That structure of the cell is self-assembled. There's not somebody directing all the components of the cell into the different compartments. So what is determining that assembly of the cell? It's laws of physics. If you take the components of the cell, the atoms, the molecules, and you mix them under the conditions in which the cell exists, the laws of physics will dictate that they will arrange themselves in the way that we see. So there's no what's called life force. That was an old theory that's been debunked. And when we talk about molecular biology and modern molecular biology, and I tell you, you know, for biologists of my father's generation, for example, my father was an agronomist. biology is a completely different subject than it is today. And that's because of the revolution that took place in molecular biology. And that revolution was sparked by physicists, among others. But the most famous example was Schrödinger. So Schrödinger, you know, in the 1930s, was about the time when quantum mechanics pretty much was established. And he was working in Germany, and Nazis came, then he denounced them, he didn't agree with them. And then he found he had to leave. He had to leave Germany. And he went to someplace like, I don't remember exactly, I think maybe UK or Netherlands or someplace like that. But he got into he didn't get along with some people. So he had to go back to Germany. Then he said, "Oh, I'm sorry." Said to Hitler, "I'm sorry, but it wasn't good enough." So he had to flee again. And so wound up, let's say, early 1940s, now about the time that the war is starting. And he's in Ireland or in the UK. And now this is the time when people are developing the atomic bomb. And he is not keen on war and killing. And in fact, he's quite frightened of the implications of the atomic bomb. So he is trying to think of, are there ways you can use the physics for more humane outcomes? And so he became interested in biology. Maybe physics can be applied to biology, and maybe there can be some life-preserving function from it rather than destructive function. So he gave a lecture in Ireland. This was in 1942, with the subject, "What is life?" So now, if we think about where biology was at this time, people had the idea that, well, first of all, they understood that cell just ranges itself. And so they thought, well, there must be some code there, like a computer code. Of course, people were inventing computers at this time. So they said, okay, so there's a code, there's a molecule that has all the instructions, the so-called genetic instructions. And the big race was on to discover what that molecule was. So in asking the question, what is life? A central concern for Schrödinger was, what is this genetic material? So he started out with this observation that living things are much bigger than atoms, quite obviously. And also living things must be ordered. They're very highly ordered. I mean, this is a big characteristic of life, right? Now, in physics, when we explain ordered systems in physics, we use statistical physics. So we have large numbers of atoms. The canonical example at this time was paramagnetism. The transition from paramagnetism to ferromagnetism. The model that explains that is based on a large number of individual atoms, each with their nuclear magnetization, and they interact with each other. And when you have lots of them, because of cooperativity, and you need lots of them, right? Because then they form these domains, and then they start to give you these ordered structures. So order is coming from a very large number of very little things. So living things are ordered, but whatever molecule it is that's giving the genetic instructions to the cell, it's on somewhere between 2 and 48 chromosomes. So just a tiny number of molecules. And yet it's producing an ordered system. So this is a paradox from the point of view of a physicist, from Schroeder's point of view, anyway. So he made this prediction about the genetic material before anyone knew what it was, and it turned out to be very accurate. And it really captures the essence of what it is about biomolecules. We're talking about molecular biophysics today. What it is about the molecules in biology that makes them special from the point of view of the physicist, or at least the physicist of Schrodinger's time. So he says the most essential part of the living cell, the chromosome fiber, may suitably be called an aperiodic crystal. In physics, we have dealt hitherto only with periodic crystals. To humble physicist's mind, these are very interesting and complicated objects. They constitute one of the most fascinating and complex material structures by which inanimate nature puzzles his wits. Yet compared with the aperiodic crystals, they are rather plain and dull. So we have this nice analogy here. The difference in structure is of the same kind as that between an ordinary wallpaper, in which the same pattern is repeated again and again and again, in regular periodicity, and a masterpiece of embroidery, like Raphael tapestry. No dull repetition, but an elaborate, coherent, meaningful design traced by the great master. So it's the aperiodicity, or we could sometimes say heterogeneity of the biological system, which is important for its function. And of course, that is what gives rise to the huge variety, the huge diversity of life that we see on this planet. It's coming from this heterogeneity. It could never come from periodic crystals. It could never come from these simple structures that physicists, at least in Schrodinger's time, worked with. So that was in 1942. And that encouraged a lot of people, physicists and physical chemists and engineers, people with that kind of quantitative background, to enter into the field in biology. And that led to the beginnings of molecular biology. And the structure of DNA was solved. And the classical central dogma emerged from these early studies. So the classical central dogma explains molecular biology in terms of the movement of information. Information starts in the DNA. That's where it's encoded. It is transmitted to the RNA. So this is deoxyribonucleic acid and ribonucleic acid. These are two molecules that chemically differ just in one position, where there's an oxygen. And then from the RNA, the information is transmitted to the protein. And in this conception, the protein does all the work in the cell. So a lot of different kinds of proteins. Each protein has a very specialized job. Each protein is a very specialized nanomachine. And the whole cell is constructed from these proteins, and these proteins ensure that it continues to function, that it preserves itself, that it divides, multiplies, and so forth. So that's the classical central dogma. It's very simple. Okay, so let's just very briefly look at the storage and transmission of information. And this was the mechanism that Watson and Crick discovered when they came up with their model for the structure of DNA. And those of you who've had a year of biology, I think have seen this. You have a chain of individual units called nucleotides each unit contains a sugar and a phosphate, and attached, and the sugar and phosphate is the same for all the units, but then each of them has attached to that sugar a base, and the base varies. This is where the aperiodicity comes in, because there are 4 different choices of bases, and we won't look at the structures, the chemical structures yet, but it's sufficient in terms of this mechanism to just name them in terms of these abbreviations A, T, G, and C. And the transmission of information comes because if you have one of these sequences, then if you take another DNA and you start to synthesize the DNA one nucleotide or 1 unit at a time, then where you have an A in the first strand, you get a T coming next to it. Where you had a G, you get a C, and vice versa. So in other words, the sequence of the second strand is determined by the first. And if you split each of these up, you can make another copy from each of them. And this is the way the information is transmitted from one generation to the next. But the transmission of information in biology is much more than the transmission of information from generation to generation. So here's a representation of the top of the structure of DNA. So this is the double helical structure. So you can follow 2 strands there, and that represents the 2 chains of sugar phosphate linkwages with the bases. And these are the chemical structures of G and G and C. And you see that they form these what are called hydrogen bonds that are represented by those lines, where you have a heavy atom, normally a nitrogen and oxygen, and one of them has a hydrogen attached to it. And the 2 heavy atoms share that hydrogen. That's what's called a hydrogen bond. And that has a favorable energy associated with it that tends to pull the 2 bases together in these cases. And so you see the structure and the complementarity of the hydrogen bonds helps to solidify that complementarity that gives us the mechanism for information transmission that we saw in the previous slide. Now, the RNA, we said, is similar to the DNA, it just differs at one position, the oxygen. But there's a much more important difference, which is not the chemical difference, but it's the form in which it occurs inside the cell. Because the DNA has 2 strands, but the RNA is just 1 strand. It shows up as just 1 strand, and that means that any one of the A's can pair with any one of the T's. Or actually, in RNA, the T is replaced with something slightly different, which is a U, which is almost the same. Again, it's just 1 atom substitution difference. But the point is that each A has a choice of any U anywhere in the sequence, also vice versa. Same with the G's and the C's. So there are a huge number of ways that those bases compare with each other, leading to a huge range of possible structures in the RNA. And then here's the protein. The protein is made up of a chain of amino acids. The amino acids have 20 different side chains. So there's even more variation that you can get from the proteins. And the proteins fold in a diverse range of 3-dimensional structures. And with these different structures, that enables them to carry out different tasks. So this is an example of one folded protein. You see that you have a linear chain like this, but it actually forms a very compact structure in 3 dimensions. So we said information storage is not just going from one generation to the next, or information storage and transmission, I should say, information transduction. The living thing also needs to exchange information with the environment. I mean, we couldn't say that something is alive if it just has a program that tells it what it's going to do no matter what happens outside, and no matter what the consequences are of its actions. A living thing is always actively interacting with the environment. So that means that our central dogma has to be more complex. So this is one way. There's no consensus on a modern central dogma, but this is one way that I would propose to describe the central dogma. We're still going from DNA, RNA, protein, but we have this yellow region. This yellow region represents the outside environment. So when we go from DNA sequence to DNA structure. So when I talk about DNA structure now, we know DNA forms a double helix, but when you get a very, very, very long chain, this double helix can curve, it can bend. In fact, in the chromosome, it's all coiled up. If you've seen pictures of chromosomes, it's not just this long linear helix. And that is something that the DNA and the chromosome folds itself in a manner that's sensitive to the environment. So it's detecting what's going on in the environment, and then it's encoding that information in its structure. So the sequence encodes the information from the previous generation. But the structure, the three-dimensional structure, is encoding the information about what is going on in the environment around the organism. So it's the DNA sequence and the DNA structure that determines the RNA sequence. And then the RNA detects what's going on in the environment. It folds accordingly. Again, it's encoding information from the environment. And it folds up into a unique structure. And then that structure plus sequence determines what the protein sequence is going to be. The protein, again, interacts with the environment that determines its structure and its function. So now, all of this should have convinced you of the importance of the three-dimensional structure of the proteins and nucleic acids. That three-dimensional structure is encoding information. And it's a mechanism for transducing information. So we can bring in a little bit of physics to address that, right? Because what decides how the structure is going to form in response to the environment? It's physics. So it's going to be the free energy of the different structures under a given environmental condition. So we can use statistical mechanics. We can take, let's say, our RNA. This example, we have an RNA. It could be a protein or DNA. And we can write down a partition function. So we calculate the energy of all the different possible ways it can fold. And then apply our Boltzmann constant, add them all together, get our partition function, and the probability of any given structure is just given by taking that Boltzmann factor divided by the partition function, which is all very familiar to us from statistical mechanics. And it leads to something that's a little bit different than the way most biologists think of things. Because, and this should be not surprising to any of us as physicists, right? The biologists sometimes think there's one lowest energy structure. course, it's not like that. Biomolecules will form a range of structures. There's a distribution. Okay, there's a probabilistic distribution. And that's actually quite important in terms of biological function. So the question, what's the Q? The Q is the partition function. So the Q is just sum over all the different Boltzmann factors for all the different states of the system. So now in this case, so there are different ways of visualizing its distribution. So for example, this slide just shows one way, which would be the base pair probability for any given base pair. We could calculate the probability that it will be present just by summing all of the little q's, that is all of the Boltzmann factors, all of the states of the system in which that particular base pair exists, and then dividing it over the partition function, summing over all the states. Ma'am? Any other questions? Okay, so that means in principle, from physics, we should be able to predict the folding of nucleic acids and proteins. So we showed a little bit about nucleic acids. So let's talk about proteins now. What we call natural selection selects for the function of a protein. So in other words, an organism will have much better chance of surviving if the sequence of its proteins lead the proteins to fold in a structure that is useful for the organism, for the survival of the organism. So that's something that is not necessarily determined by physics. But once you have the sequence, as it's been selected, then in principle, you should be able to predict the structure from physics. Now, when it comes to evolution and mutations, then going from one generation to another, an organism can adapt to the environment through mutating the sequence of its DNA, and that will eventually lead to changes in the sequence of the protein and changes in function. But actually, the organism can respond much more rapidly at the level of the RNA, because the RNA is just synthesized temporarily and then it's disposed of. So mutation, so-called, can occur much more rapidly at the RNA level. So for example, the RNA can splice itself, it can cut out a part of itself and then splice the remaining parts back together. And this will lead to a protein with a completely different sequence, a completely different protein. It can do that in response to signals from the environment. So that's actually much more rapid response than just mutations at the DNA level. So, the connection, of course, between the sequence and the structure is thermodynamics or statistical mechanics, as we said. But the thermodynamics doesn't always link the function. What do I mean by that? There may be the lowest free energy structure may not be the one that actually works best for what the cell needs. And there can be cases inside the cell in which metastable structures may be the functional structures. So we always have to think of this process inside the cell in a very dynamic way. We have to consider not only the thermodynamic equilibrium state, we have to think of the kinetics. We have to think of the folding rates and compare those folding rates with the biological processes that are taking place and the functional requirements of the cell in order to predict what's going to happen. So all that having been said, at least we could predict the thermodynamics part, the equilibrium part, let's say from physics, right? But to do that, we need the forces, we need the potentials that are driving this folding. So what are those? And they are classified historically, not really by physicists, but according to people's experience. So on the one hand, we have some of them that as physicists we would recognize, like electrostatic forces. Conformational entropy. Conformational entropy refers to the entropy that is associated with the with flexibility in terms of the shape of the backbone, for example, which is something which is entropically advantageous. If you have a structure that allows for some dynamics in a segment of the structure. Then we have hydrogen bonds, which we saw in the earlier slide. The hydrogen bonds are kind of from a physicist's point of view, they're kind of from the point of view of physics students, they're a source of anxiety, hydrogen bonds, because they aren't just arising from one force. The chemists tend to teach their students that there's just an electrostatic interaction because the heavy atoms are slightly electronegative and the hydrogen is slightly positive. And that's a part of what's contributing to hydrogen bonds. But actually, there's a component of hydrogen bonding which is covalent, which involves sharing of electrons. And all of that is very much a current area of research in terms of really pinning down what all the contributors are to the hydrogen bond and what the relative contributions are. Then there's van der Waals interactions, which you might remember from your chemistry. So van der Waals interactions arise where you have, well, they tend to bring large surfaces together. This is very important for biomolecules because they're large molecules and they have very large surfaces. So that can lead to very large van der Waals interactions. The van der Waals interactions, again, they are something that were invented by chemists. And in terms of the physical origin, they're actually a combination of a number of different physical forces, including quantum mechanical interactions, and we usually say electrodynamics. Van der Waals forces are considered to be very important for something called hydrophobic interactions. Hydrophobic interactions, again, it's not defined by the fundamental physics, it's defined by the observation the oil and water separate. And whatever forces, actually the forces that cause that are statistical mechanics. But they're not fully understood. Yeah, but they're very important in biology. They're very important in protein and nucleic acid folding. And those forces are called hydrophobic forces. But in fact, they're a combination of all these other forces. There's electrostatics involved, there's hydrogen bonds involved. And again, that's an active area of research. And we're going to look at a moment in disulfide bridges. Disulfide bridges are a particular kind of chemical bond in proteins that have a big impact on their folding. So for a long time, from the origin of biophysics, we could say that predicting protein structure for first principles was the holy grail. And there were two basic approaches based on physics or based on homology. So homology means you look at the sequence and you try to find another protein that has a similar sequence for which you know the structure. And you try to use that as a means of predicting the structure. And there were huge armies of people all over the world, physicists, physical chemists, working on trying to solve this problem, using lots of different methods, with different combinations of the physics-based and the homology-based methods. And some people consider that the problem has been solved now. We'll get back to that later. But first, let's just make sure that we all have a little bit of the basics about what a protein is. So a protein, like a nucleic acid, is made up of a chain of small units, the small units of the amino acids. And the amino acid has a nitrogen, a carbon, and a second carbon. The second carbon is linked to an oxygen. So those three atoms make up what's called the backbone for the protein. Each subunit, each amino acid has these three elements: nitrogen, carbon with some kind of aliphatic. A side chain is actually different side chains for each amino acid is connected here, and then the carbonyl. And you see that we have another nitrogen, another carbonyl, nitrogen, carbonyl, so on. So if we look at the backbone, so for the moment, ignoring the side chains, so we haven't shown in this slide, then for each amino acid, there's limited degrees of freedom in terms of the structure. So you've got 3 bonds in the backbone, and for one of those bonds, this one here, which is called omega, which links the carbonyl to the nitrogen, there's a partial double bond character, and that means a rigid bond. So we can have rotation about this bond, which is called phi, and this bond, which is called psi, but not about omega. So we have 2 degrees of freedom, 2 degrees of rotational freedom per amino acid. Now, if it was just like this, no side chains, then we would have quite a bit of freedom, but we don't have complete freedom, because if we rotate about those angles at phi and psi, then the backbone atoms have other atoms attached that start rotating like this, and in certain configurations they clash with each other. We have steric clash. So not all of the 2-angle space here is actually accessible. So we ignored the side chains in the last slide. There are 20 possible side chains in naturally occurring amino acids, and this is all the 20. And if you haven't had your biochemistry before, then I'm not going to make you cover all these. But the most important thing about them is that we can categorize them according to their physical properties. So we have a set of them here. The ones I've highlighted in purple. These are called the aliphatics. So all of them, you see, they have this nitrogen, oxygen. But then the side chains here are just carbons. Or here we have in a couple of cases sulfur. So carbons are non-polar. They don't have a charge. So those side chains will lead to a oily surface, and those side chains end up going more. Those are called the hydrophobic side. Then we have here what are called the aromatic side chains. They have aromatic rings, and those also tend to be hydrophobic. And they can stack on each other. There's a lot of energy from these rings stacking on each other. And then we have these red ones. These red amino acids, there are 4 of them. They have charge groups here. Like here you have carboxylic acid that's negatively charged. Here you have what's called guanidine, all these nitrogens, that's positively charged. So of course those amino acids will tend to interact with the negatively charged amino acids. So positively charged, like arginine, will tend to interact with the negatively charged, like glutamic acid. And then we have the blue ones. The blue ones are polar. They don't have a net charge, but they have these heavy atoms on them that are electronegative or electropositive. So they have what's called partial charge. And those that interact with microbes, with each other or with water. Now, since we have only 2 degrees of freedom per amino acid, we can characterize the whole of the backbone for the amino acid from these 2 degrees of freedom. So our accessible conformational space, we can map. We can think about this being something like your phase space, the phi size space, the different configurations of the protein. And there, we said before, they're restricted by steric blocks. So we could plot those and show the regions of conformational space that are accessible sterically. We can do that for polyalanine. Alanine is the amino acid that has no side chain. So that's got the minimal steric blockage. And then we can add the potential functions. We can add the forces that we talked about, the electrostatics and the van der Waals forces. So, and that will further limit the accessible range. So let's just have a look. That kind of map. This kind of map is called the Ramachandran map. After the guy in India who was the first to present this, Ramachandran, he was a famous biophysicist, and his son, my classmate in Berkeley, he was studying subject of the last talk. He was studying plasma physics, and remember we were having a conversation over coffee and about our families, and then said, "Yeah, my father was a biophysicist," and he started describing what he's doing. Your last name's Ramachandra. Same guy. So Ramachandra graph is, as I say, it's a way of mapping the protein conformation. So you have phi can go from -180 plus 180, psi can go from -180 plus 180. And so for each amino acid, we can map where it is on this chart. And for a protein, we'll have a number of different spots representing different amino acids. And in principle, we have all the information about the protein vector structure on that map. And what we find is that simply based on steric considerations, there are only a few regions of this map that are accessible. So we have this region down here. So we have the solid lines and the dotted lines. Those depend on what we assume as the van der Waals radius. As the larger you assume the van der Waals radius, the smaller these accessible regions become, because there's more steric constraint. So we have this region down here, which it turns out is characteristic of a type of protein secondary structure we call the alpha helix. And then up here, we have another region which is characteristic of another secondary structure called the vega strand. And then actually this area here, so what's called the left-handed alpha helix, and it's actually really occupied. So now that map was just based on steric considerations. Now, if we calculate potential energy, and we include our electrostatic energy, we include our hydrogen bonding energy, we include our van der Waals energy, and then we also put some energy associated with any distortions in the bond lengths and the bond angles. We can treat those as being harmonic potentials. And then we add all that up. And we get a map that looks like this. So now we have contours, which are energy contours. And we see again, minima in this alpha helix region, above that, the beta strand region, left-handed region here. And now we have a map here from I think it's 1981. 1981, there were just, I don't know, a few dozen protein structures. And the data from those structures is plotted on this map. And you see that the points for the individual amino acids map pretty well onto that Ramachandran energy map. And this one is updated a little bit to 2003. And here we have just probability density based on the databases. And again, we'll see maybe these two potentially. Now, what we looked at was for alanine, or just a generic protein, let's say with a mixture of all the 20 amino acids. But there are some differences in the Ramachandran map, depending on the side chain. In particular, for glycine, which has the smallest side chain, it's just a methyl group. You see that you have a much bigger minimum here on the right. And also you have more data points that are going outside of these three major minima. And proline is very exceptional. See, the proline is very highly constrained, but the amino acid before the proline has a unique distribution as well. And we'll come back to that too when we look at some of the structures, because we'll look at it in terms of the structure of the proline side chain. So protein structure is built from modular elements, and we have a handful of motifs of what are called secondary structures that we see repeating themselves in proteins. And it's essential to get familiar with these mainly alpha helix and beta sheets, which we already made reference from chocolate map. And then there's some variations here with the 310 helix and the beta turn. So let's just very quickly see what those look like. So first, we have the alpha helix, and it forms a helix, which you can see here. But here I'm showing it schematically without the helix, just looking at the side of it, just to point out that there's a hydrogen bond going from amino here to carboxylic acid here, and it's separated by 3 residues. And the helical structure places those two, the nitrogen and the carbonyl, next to each other to form the hydrogen bond. And then when you have a whole series of these turns, there's a very small dipole moment associated with each turn. And when they're all aligned like this, then you get a significant dipole moment associated with the alpha helix. The 310 helix is a tighter helix. You have hydrogen bonds going between amino acid, and the amino acid removed by two residues. And then the beta turn is very important because that generally is in regions that are linking in between alpha helices or beta strands. And that's where the direction of the backbone changes. So the beta turns can have a number of different configurations. But one thing I want to point out is that beta turns tend to occur more with some amino acid side chains than others. And in particular, this proline, this is the proline side chain. You see it's very exceptional compared to the other side chains. And it's very highly constrained. And that's why it often occurs in beta turns. So beta turns consist of 4 amino acids, and the second one is usually proline. Okay, so we said protein structure was always the holy grail, predicting protein structure, and then about 5 years ago, we had this big breakthrough from deep learning, the so-called AlphaFold, and then people decided, or many people decided, well, our AI has solved the protein folding problem. I mean, this was one of the big events you might remember that was seen as proof that AI now has come of age because it solved this very difficult problem. So the event really that triggered this was a competition. There was a biannual competition, long been a biannual competition between the different groups trying to predict protein structure. And in 2019, this competition was won by this deep learning algorithm. 2020, I guess. So, but in order for the AlphaFold to get this up to almost 90% accuracy of prediction, it needed a learning set of 19,000 structures in the protein database. So each of those 19,000 structures required a team of at least 3 or 4 people working minimum 2 to 6 years, let's say, with a lot of money to solve those protein structures. So that's something to keep in mind. But there's still some issues because this algorithm assumes that the structure is static. Most of these structures were solved by crystallography. And in crystallography, we freeze the structure. But we've been saying that proteins and nucleic acids, they form dynamically. They're in motion all the time. And they form a distribution of structures, not population distribution, not just one structure. So, but maybe more importantly, there's the when a protein binds to another biomolecule, it very often changes structure. And this is where, as we said before, the function of the protein or the nucleic acid comes in. It's all about the interactions. So it's interactions between the proteins, proteins with nucleic acids, proteins with DNA, proteins with RNA, RNA with DNA, chromatin, where you have proteins and DNA complex together. And in so-called ribonucleoproteins, like the ribosome, where you have a complex of RNA with proteins. And you also have interactions between these large macromolecules and signaling molecules. They're called signaling molecules. Small molecules like vitamins, metabolites, drugs basically come in and hijack those roles. And these small molecules can lead to big changes in the structures of these large molecules. So these interactions are key to understanding how enzymes act, for designing drugs, for synthetic biology, for understanding the regulation of gene expression. And core issue in this process of interactions between biomolecules, involving biomolecules, is specificity. Specificity meaning, how does each protein or nucleic acid choose its binding partner from all the other molecules in the cell? And the specificity is really extraordinary. And if we believe this static picture that we could just look at the structures of the individual proteins and they're static, and they just fit together like Lego. It'd just be a matter of this shape fits together with that shape, but that's not the case. It's been this misconception dates back to a model called the lock and key model for enzyme recognition. It goes back to the 19th century. It's attributed to Emil Fischer, who was the son of a brewer. He was playing around with his father's yeast cultures, and he extracted enzymes from them. And then in the laboratory, he used these enzymes to ferment different sugars. And he found that the enzymes had specificity. So one enzyme would prefer one sugar, and another enzyme would prefer another sugar, and so forth. And so he suggested, in order to explain this, that the enzyme and glucoside have to fit together like lock and key for this chemical effect. And then he went on to say, still more important, though, appears to be the proof that the previously assumed difference between the chemical activity of the cell and the mode of action of chemical reagents is factually non-existent. And this actually comes back to something we talked about before. Why did he say that? Why did he emphasize this? This was the fact that he was able to have the reactions take place outside the cell. He said that because in those days, people like Louis Pasteur still believed in life force. So they still believed that enzyme reactions could only take place inside the cell. And this was disproved. But this lock-and-key model also didn't really survive with time. So in 1958, Dan Koshland proposed what's called the induced fit model. So here's the figure from his original paper. So these bars represent the active site, the so-called active site of the enzyme. That's the site where the substrate sits and where the reaction takes place. And you see that it's flexible and it shifts around. Now, these are the so-called substrates. So Koshland was looking at enzymes that cleave proteins. And he was noticing that they would cleave only proteins that had certain amino acid sequences and not others. And he was trying to explain that specificity. So he said, well, there could be what he calls your non-substrate. That would be a protein, for example, that has the wrong sequence. It can bind to the enzyme. It binds to the enzyme, but it doesn't cleave it. Okay, why is that? So his suggestion is that when the substrate binds, it induces this structure in the enzyme itself. And it's only certain substrates which will induce a structure that puts this catalytic group next to the reactive component substrate, and they can actually catalyze the reaction. So he said this set of postulates is called the diffuse fit theory, and the idea of fit is obtained from the law theory that the fit in this case occurs only after the changes induced by the substrate itself. And then in the 1990s, people proposed another model for how this, how the ligand induces changes in the conformation called conformational capture model. So this model actually brought in a little bit of thermodynamics, at least very basic thermodynamics, the Boltzmann distribution, acknowledging that the protein is not just sitting in one structure, it's sampling different structures. And the proposal is that when the protein samples the structure that fits the ligand jumps in and it interacts and it holds it. It captures that conformation. So that's called the conformational capture model. It was very much inspired by some of the early studies using NMR, which were in solution, unlike crystallography. And with NMR, you could see that there are certain regions of the protein that were a little bit more flexible than others, and it was observed quite often that you added the ligand, you added the substrate, then that region would become less flexible. Okay, so I think we're getting close to the end of our time for our session today. It's not the ideal place to have a break, but I'm going to next, the models that I've talked about, this induced fit model and conformational capture model and key-lock model, these are cartoons. They don't give us any basis for quantitative predictions. But there is, fortunately, and it goes back a long way, a model, a very simple quantitative model for the interplay between ligand binding and protein conformation, which is called link equilibrium. We'll start next time with going through this model, which was actually described in this text from the same year that Koshland published his induced fit model by scientists in Harvard and Iowa. So before we leave, I want to ask you guys, I mean, you don't have to do this, but it will be ideal if you could. To download a program called Chimera X. Chimera X is a program, open source program that you can use to visualize biomolecular structures. So let me just write it up here. And we'll try to have a session next time where we actually move the structure around and play with it. And in order to be able to make use of that program, we need the structures themselves, and we can get the structures from the protein data bank, or it's called the PDB. So you can do a Google search for RCSB PDB, and actually there's a lot of educational tools on that site, very basic tools for
protein and nucleic acid folding. And just very quickly, let's see if I can share my screen just to show you. Okay, stop sharing, and then I'll share the window for UCSF.
Okay, so this just shows you the interface for the program. And we're going to look at some structures, we're going to visualize them like this, practically around this window and display them in different ways. So the files have the extension. So the program can read files with this. So I guess we're out of time now. Are there any last questions for the session for today? So is the level of this right for you guys in terms of biology? I know that the physics here is very simple for you guys. Is the biology getting boring for you, or is it getting to be too much? I mean, but am I assuming too much or too little? Okay. All right. Okay, so so our session tomorrow is the 2nd in the afternoon, right? So it would be at 3:30. See you then.
This is in the building.